Hey, thanks for coming. My name is Daniel Haas. I'm a postdoc in Jim Hurley's lab. We're in the biochemistry department at the University of Washington School of Medicine in Seattle. Neither me nor any of my authors have any financial interests to disclose. So we're going to talk about the idea that gluconeogenesis can happen in the mouse retina and eye cup. We found that out by infusing 13C labeled succinate. I will explain what all of that means now. So what is glycolysis? Glycolysis is taking a glucose molecule, one of the primary energetic intermediates that fuels metabolism throughout the body, and it breaks it down. So glucose is represented by this series of six circles at the top of the diagram I'm showing. This is the glycolysis pathway and how it enters into the Krebs cycle. Glucose gets broken down and energy gets produced. That's the simple description of what happens. Many of the steps in glycolysis, however, are reversible. And so sometimes different tissues in our body, including especially the liver and the fasting liver and muscle, sometimes they want to make glucose rather than breaking it up for energy. And they make glucose for other cell types that are around them, just to make sure that some cells that have high energetic needs have a constant supply of energy to fuel those needs. And many of the steps in glucose breakdown are reversible. That's indicated by the double-headed arrow. But then some of them are also irreversible. That includes phosphofructokinase and hexokinase and pyruvate dehydrogenase, for example. And so sometimes you need a different enzyme in order to allow those reverse reactions that make glucose to actually take place. And so the enzymes that are critical for gluconeogenesis include, but I don't think are limited to, phosphoenolpyruvate carboxykinase, glucose 6-phosphatase, and fructose bisphosphatase. And those are all represented in the red arrows here. So those are the critical gluconeogenic enzymes. And we're really going to focus in on the activity of phosphoenolpyruvate carboxykinase, because that's really the first committed step in gluconeogenesis. That's taking the molecule oxaloacetate from the TCA cycle, diverting it away from normal metabolism to make phosphoenolpyruvate, which can then go into a gluconeogenic pathway. So these enzymes, these special gluconeogenesis enzymes, are only expressed in certain tissues, and we really wanted to find out if we could sort of detect those activities and what tissues they might be detected in. And so what we did was we took mice, and these are freely moving, awake mice, and we can infuse a labeled metabolite directly into their bloodstream. Here I've indicated that it's 13C succinate, and that's what we infused. And U13C really means uniformly labeled 13C. Succinate has 4 carbons, and so all 4 of those carbons are going to have an atomic mass of 13 instead of 12. And so we can actually then infuse that labeled succinate, let it go throughout the body, be used in different ways by different tissues, whether that be for gluconeogenesis or sort of more conventional oxidation reactions. And it'll be used in retina, eye cup, liver, lung, yada, yada, yada. We've taken all these tissues and we've put them on liquid nitrogen to flash freeze them, and we can analyze metabolites using this method, gas chromatography linked mass spectrometry. And that allows us to separate out different molecules by volatility and then the molecular weight of those molecules by the mass to charge ratio. And the mass to charge ratio really tells us whether or not that molecule is heavy and how heavy is it. So if there's one heavy carbon on succinate, for example, we'll have it as M+1. If you look specifically at these series of 5 peaks here, you can see there's this M+1 would be this slightly shorter peak after the tallest peak in the mass spectrometry cartoon. And based on the different levels of these isotope logs, we can really determine what kind of metabolism that 13C is going under. So 13C succinate, we can tell what pathways it enters into. And so this is just to show what happens in blood, retina, and eye cup after we've infused that 13C succinate into the mouse's blood. And so the red line in the leftmost figure indicates the fully labeled succinate in blood after we've infused it. We can see before the infusion, it's practically non-existent. And after the infusion, it drastically increases and then really quickly decays in blood, which shows that our infusion is essentially successful. And the scale of the succinate that we infused kind of silences the background succinate that was already in blood. And so you can see that in kind of the black line, relative to the black line, the red line is gigantic. So all of the succinate that the tissues are going to see is going to be 13C labeled succinate. And then when we look specifically in the eye cup and the retina, those are the tissues that we dissected out and flash froze, we can look at fractional labeling of succinate with 13C. And so I've separated out here the different isotopologues, that is, the different masses of succinate, whether or not they were labeled with 1 heavy carbon, 2, 3, or 4 heavy carbons. And you can see that both in eye cup and retina, the major isotopologues are either unlabeled succinate that was already there before we infused anything, or the fully labeled succinate that the tissues both get from blood. And the eye cup clearly gets a lot more fully labeled succinate than the retina. This 0.6 means that 60% of the succinate pool in the eye cup is labeled by 13C. And in the retina, it's only roughly 8%. And so that's really important to note as we move forward, that different tissues will accumulate different amounts of 13C labeled succinate. So what is the hypothesis? How are we going to determine whether or not gluconeogenesis happens using succinate? It just so happens that succinate in the Krebs cycle is a precursor to oxaloacetate. And if you remember, oxaloacetate itself participates in the reaction that forms phosphoenol pyruvate in the first committed step of gluconeogenesis. And so these filled in purple circles represent fully labeled carbons that are initially on the infused succinate, make their way over to 4 fully labeled carbons on oxaloacetate, and then in the process of the Pepsi-K reaction, which turns oxaloacetate into PEP, it loses one of those labeled carbons, and so you'll see fully labeled PEP is actually M+3 labeled. And so our hypothesis is really that we're going to see, after we infuse labeled succinate, an increase over time in labeled phosphoenol pyruvate. That's indicative of gluconeogenesis happening. And that's exactly what we see. When we look in retina and eye cup, we look specifically at M+3 labeled PEP to see that heavy labeled PEP that came from succinate. And it's increasing over time in both the eye cup as well as the retina. And that increase is both present in the PEP fraction, PEP pools, as well as the 3-phosphoglycerate pools. That's even further upstream glycolytic intermediate, which shows that 2 different molecules in glycolysis are both accumulating heavy label that originated from succinate. So that shows gluconeogenesis is happening in both these tissues. And when we compare the amount of M+3 labeled pyruvate at a single time point to the amount of M+4 succinate that actually entered into the tissue, just to give us a relative metric of how much gluconeogenesis is actually occurring, we can see that the gluconeogenesis in the retina appears to outstrip gluconeogenesis in every other tissue. And it's also present in eye cup, but surprisingly, tissues we know to carry out gluconeogenesis, like the liver, aren't really doing it. And we think that has to do with the fact that these mice were not fasted or starved before this assay was carried out. And if they were, we might see the liver does a lot of gluconeogenesis, which has been confirmed previously. And we also found that when we take these retinas and eye cups and we put them in a dish and incubate them in a dish with 13C labeled succinate, we don't see any accumulation on M+3 PEP or 3PG. And that really just goes to show that the environment in vivo and ex vivo is really critical in dictating whether or not this gluconeogenic reaction is going to occur. So the differences in the environment that we've actually really been considering, if you look at this reaction diagram, are the fact that most cell culture environments, including the one we've used, utilize CO2 to maintain cell culture pH. But CO2 happens to be a product in the Pepsi-K reaction. So it could present a form of product inhibition, which prevents PEP from actually forming from oxaloacetate. So we think this might be the reason we don't see the reaction occur ex vivo, and we're going to try to confirm that just by using a pH or a CO2-free medium in the future and do these same ex vivo incubations and see whether or not we get 13C labeling on phosphoenolpyruvate from succinate. But overall, we still have this really surprising result that the retina carries out gluconeogenesis. And so we still don't know exactly why it is, but we think perhaps some cells in the retina are storing energy in the form of glucose as a way of allowing other cells, which might have high energy demand, to get energy when they need it. If vascular supply can't meet their demand, then perhaps other cells in the retina already have glucose that they can provide to compensate for a lack of vascular supply. So this is still a hypothesis, of course, and we need to confirm that gluconeogenesis is happening in other ways. But we think it's a pretty strong argument that we're making that it is happening. And so I'd like to thank everyone in my lab for really just helping me out just by talking and throughout this pandemic being really awesome people. I've bolded the names of the people that have helped me specifically in this project, either by helping me dissect or perform surgeries on the mice to get the catheters installed. And I also wanted to note that both Caitlin and Celia, they're highlighted in red, are presenting posters at Arvo. And I think you should check them out if you have the time. I'd also like to thank the NEI for funding support, of course, and my mentor, Jim, for just being an awesome mentor. And so with that, I just wanted to invite you to ask me any questions you might have. And thank you for stopping by.